Grapeseed extract reduces Anco biofilms.
In this video, I'm going to be talking about Anco biofilms, which is similar to biofilms that bacteria form. But in this case, I'm going to be talking about also grapeseed extract, and you could possibly find that in supplement form. So Anco biofilms, they're basically highly adhesive structures formed by cancer cells, and they help these cancer cells survive chemotherapy and also enhances metastasis. So it's important to degrade those biofilms because metastasis is pretty much the growth and the increased numbers of cancer cells. Okay, so they actually involve dense extracellular matrix fibers, including collagen, hyaluronic acid, laminin, and proteoglycans. So ECM is short for extracellular matrix. And these oncobiofilms are especially noted in breast cancer, colon cancer, glioblastoma, and pancreatic cancers. Now, grapeseed extract, GSE for short, actually controls oncobiofilm formation by affecting the pathways that form or help modulate oncobiofilms. And GSE for short has catechins and gallic acid with strong antioxidant, anti-inflammatory effects. Now, the anti-inflammatory part is critical, super important to reduce the inflammation because oncobiofilms, they produce inflammation if they continue to be ongoing and they are not degraded. And the inflammation can actually degrade or block the vitamin D receptors. It's critical to reduce oncobiofilms because ongoing inflammation can decrease and downregulate the vitamin D receptors. So that means that things like zinc finger proteins will not work very well because the zinc fingers are attached to or are a part of the vitamin D receptor. And so that goes to show that the vitamin D receptors has interactions with zinc, and zinc is important for the immune system as well as the thymus. And thymus needs zinc because the thymus is where the T cells train. So because of oncobiofilms and the inflammation that continues, not only are the oncobiofilms protecting tumors from immune cell attack or macrophage phagocytosis, but indirectly because of the inflammation and the effect on the vitamin D receptors and zinc, that inflammation from oncobiofilms are also reducing T cell numbers, therefore less T cells to attack cancer cells, because if zinc is not getting or if zinc is not being available for thymus, or the thymus is not getting the zinc, then the thymus is not creating as many T cells to battle or fight cancer tumors, allowing tumors to metastasize. So grapeseed extract may affect oncobiofilms and cell adhesion. Uh, GSE has been shown to downregulate integrins AV beta 3 and A5 beta 1, and also reducing E-cadherin mediated compaction of tumor clusters. This weakens cancer's ability to maintain dense biofilm-like structures. Now that's great, so that the the biofilms are not too strong. Because what happens is when the macrophages, when they try to engulf the biofilm or oncobiofilm, it actually can't do so, and then ends up making the biofilm thicker and stronger and tougher to break through, which actually is a self-enforced feedback loop, because the stronger the biofilm is, the more the macrophage tries to engulf it, uh, meaning that it just makes it even thicker. So that's why it's better to leave the oncobiofilm alone, or to prevent macrophages from making it thicker by attempting to engulf it. The grapeseed extract prevents that from happening by not allowing the oncobiofilm to even become strong in the first place. Because if it is, then that leads to ongoing inflammation, and then the vitamin D receptor becomes downgraded or disrupted because of the inflammation, and other things uh, that are dependent on vitamin D will not be able to work either, such as at the DNA chromatin level. The DNA is inside the chromatin. The vitamin D receptor actually interacts inside the chromatin with the DNA for immune functions. Okay, so not only does grapeseed extract reduce oncobiofilm formation, but it also supports and helps T cell infiltration of the cancer cell tumor environment, because grapeseed extract interferes with ECM production, because the ECM, or the extracellular matrix made of collagen, hyaluronic acid, ECM deposition, formation is created via TGF-β and SMAD signaling, and GSE reduces this TGF-β SMAD, therefore lowering collagen and reducing CAFs as well. I've made other videos on CAFs. So basically, CAFs, they help tumors grow by transferring or giving them exosomes. So tumors can receive exosomes from CAFs, and tumors actually even recruits CAFs, short for cancer-associated fibroblasts. Uh, So, grapeseed extract is having a triple hit on cancer tumors, because it reduces biofilms, it also helps T cells infiltrate tumors, especially at the ECM, and grapeseed extract also reduces cancer-associated fibroblasts. So the grapeseed extract helps the T cells, because once the T cells migrate through or enter the extracellular matrix of the tumor, um, the T cells must physically move through dense ECM made of, as I explained earlier, collagen, fibronectin, laminin, and proteoglycans. But if that is too thick and rigid, then I've said this in my previous videos many times, that the T cells, they can't really migrate through. So the grapeseed extract can really help the T cells, because the grapeseed extract reduces the collagen, the fibronectin, etc. So T cells, in order to penetrate dense tumor stroma, they release enzymes such as MMPs, MMP2 and MMP9. These cut collagen and ECM proteins to create migration paths. Those are the paths that T cells need to migrate through to get into the tumor to attack cancer cells. Of course, it could be wise to help out your T cells with grapeseed extract. So, in case the ECM is super thick and super rigid, and the MMPs that T cells release are not enough to cut through a dense ECM, it would be great to help them out, to help the T cells out with grapeseed extract. So that's it for the video. If you want more future videos, like, comment, share, subscribe, and I'll see you in the next video.